So welcome back to another video tutorial from Biotopic Guru, and in this video lecture we are going to talk about ABO blood group. The plasma membrane of RBs contains many antigens. In presence or absence of antigen, that is blood is categorized different blood group based on its antigen. Okay, so ABO blood group, there are 24, at least 24 blood group there in presence or absence of various kind of antigens, that is the 24. Okay, so 24 blood group, at least 24 blood group. But similarly ABO blood group, that is including blood group A blood group, B blood group, AB blood group or O blood group and O blood group. Okay, A blood group, B blood group, AB blood group and a O blood group. In ABO blood, that is based on the ABO antigens. That means ABO antigens presents or surface or presents on the plasma membrane of RBC. It is the A blood group, it is the plasma membrane or it is the RBC. This is the something like that, the structure RBC. A blood or A antigen that is present on the RBC. A antigen. Similarly B blood group, that is B RBC, that is B blood group in RBC contain the B antigen on the surface of RBC. This is the, and AB, it is the both type of A and B contains the RBC. Both type of AB antigens present. In case of O blood group, that is the O blood group, they cannot be contains A blood group or B blood, A antigen or B antigens. It is the O blood group, it cannot be contains or attached to with the A antigen or B antigen, only I molecule. Okay, it can have a I molecule. I molecule is the H molecule, it is the representative I, it is similar to I, that it is the H molecule, that is it can be found in the every individual in the blood group. Okay, A contains A blood group contains A antigen with the I molecules, H molecules, that is B contains B antigen or and a B antigen or H molecule. AB blood contains AB, both A and B antigens with the H molecule, and O antigens that contains only H molecule, cannot be contains A antigen or B antigen. Okay, so RBC, it is the I molecule, it is present H molecules, it is present in every individual from blood group. It is the antigen, presence of antigens, A antigen, B antigen, C antigen, A antigen, B antigen and AB antigens. It is the genotype, it is expressed. IA, it is the allelage. I means allelage, that is allelage present in the homologous series IA, or it is IA or II. This is the H molecule. And similarly B blood group, that is B antigen in B blood group, B antigen is present with the I molecule in its genotype IB or II. Okay, so AB antigen contains IA, IB with the H molecule, and O blood group cannot be contains both A and B antigen, but it contains I molecule. Okay, so ABO blood group contains ABO antigens. ABO antigens, that is the complex oligosaccharide. ABO antigens, that is complex the oligosaccharide. That means the carbohydrate that is in the form of glycoprotein or glycolipids. Complex ABO antigens contains oligosaccharide, complex oligosaccharide, that means carbohydrate, complex carbohydrate, the oligosaccharide in the form of glycoprotein or glycolipid, that is the carbohydrate plus protein is glycoprotein, carbohydrate plus lipid is called glycolipids. Okay, and IA and IB is the allelage, that is help to production of A antigen and B antigen. The IA, that is production of A antigen, IB, that is production of B antigen and A antigen. Okay, so it is the complex oligosaccharide antigen, ABO antigen, that is the similarly chain manner Please see this. It is the protein or lipid, that is the, it is the complex protein or lipid portion, a lipid that is such as ABO blood group, that is A antigen, B antigen, chain is similar but difference is arises. Please see this. It is the glucose, galactose, N-acetyl glucosamine, galactose and fucose. In this case it is the complex oligosaccharide. Okay, our complex oligosaccharide chain, whenever we called the A antigen, if another addition of the N-acetyl galactosamine with the help of an or catalyst of the N-acetyl galactosamine transferase. Okay, is the N-acetyl galactosamine transferase. It is the addition of another galactosamine, N-acetyl galactosamine, it is called as a antigen. Similarly, B antigen, when we call the B antigen, this chain is similar, in this chain is similar, but difference is arise that in A antigen contains the N-acetyl galactosamine, but difference is in B antigen no N-acetyl galactosamine is present, only galactose. Okay, please see this. It is the glucose, galactose, glucose, N-acetyl glucosamine and the galactose and the fucose. Okay, so in this case, in this chain only the galactose is present with the linked with the another type of galactose. It is the galactose, that is the galactose in this case. Please see this. It is the galactose, it is the galactose in this chain, galactose, that is addition of another galactose. This is the galactose with the help of an or catalyst of the enzyme, that is the galactose transferase. Okay, so it is called as B antigen. You can remember this, when a chain that contains the another, that is addition of N-acetyl galactosamine, is called as a antigen, and when the only the galactose is present in this chain is called as a B antigen. B antigen. Okay. So it is called as A antigen or a B antigen. In the difference is the, it is contains the N-acetyl galactosamine and it is contains the only galactose, and it is glycosidic linkage in between galactose and N-acetyl galactosamine, that is alpha 1,3 glycosidic linkage, alpha 1,3, and in this similar, that is alpha 1,3 glycosidic linkage, it is the galactose. Okay, so it is the A antigen and B antigen is formed, which is present on the plasma membrane of RBC as ABO antigens. Okay, so in A blood group, that is the IA, is codominance with the codominance so with A blood group, A antigens. Similarly, IB, that is codominance with B antigens, but IA, IB are completely dominance to the H molecules, that is the completely dominance with H molecules, that is the hierarchically it is the symbolized IA equal to IB is greater than H or I, it is H molecule or I. Okay, so I H molecule can fails to specify A or blood A and B blood group. Okay, I molecule, it is, so it is called as completely dominance over the H molecule. It is called as IA is allele form, that is production of A and both type of A and B antigen. IA, that is production of A antigen, IB, that is production of B antigens. Okay, so blood plasma that is interact with the B antigen, A antigen or B antigen. Blood plasma also contains antibody if the A antigen or A blood group contains A antigen of A antigen surface of RBC. Antigens contains the surface of RBC A antigen, but in the plasma contents in RBC, that is the anti B antibody. In A blood group, that is contains the A antigen on the surface of RBC, but in plasma anti B antibody contains. Similarly, in B blood group contains B antigen on plasma membrane, that is contains B antigen on the plasma membrane of RBC, and the contains A anti-A antibody in the plasma membrane. And the AB blood group cannot contains both A and B blood group. Okay, both of A and B antibody, that is the cannot be contains a AB blood group anti B antibody or anti A antibody in the AB blood group, only AB blood group. But O blood group, that is contains the both type anti B antibody or anti A antibody. So it is called as a universal donor, or it is AB blood group is contain, it is called as a universal recipient con, it is called as universal recipient, and O blood group is called as universal donor. So differentiation is arising that O blood group that gives to all blood group, that is gives to all blood groups to donate the cell, that is the blood cell to give the another blood group, that is it is transfusion. O blood group can donate to another blood group, that is in case of A blood group can be received from the O blood group, and B blood group can be received from the O blood group, and AB blood group can be received from the O blood group. But reverse is cannot, it is impossible, reverse is impossible, that O blood group cannot be received in case of A blood group, B blood group and AB blood group. Okay, so O blood group, it is called as a universal donor, it is only can be donated, cannot be recipient, it is only received in O blood group. Okay, it cannot be received in A blood group, B blood group and AB blood group. But AB blood group can be recipient in such a way that A blood group, B blood group and AB blood group. It can be received A blood group, B blood group or AB blood group or O blood group. It can be received, it is called as universal recipient. But the question is arising that why should A blood group, A individual cannot receive from the B blood individual? Okay, so what happen in this case, if it is the A blood group and it is a B blood group, why should it is impossible B blood group cannot donate to the A blood group? It is impossible because of A blood group, that is contains the A antigen in the plasma membrane of RBC and a B antibody in the plasma. Okay, this is the RBC of a blood cell. It contains the A antigens and the anti-B antibody contains, and similarly in this such a way that is contains B antigens in the on the plasma membrane of RBC, B antigens and a anti-B antibody, anti-A antibody. Okay, in such a when B blood group can donate to the A blood group, the maximum time blood is clumping, and many times the blood is clumping, destruction of RBC, and the person will be severe and may be death. severe may be death. It is called as a blood clumping. Because of antibody and it contains the antigen A antigens and it is the A antibody can be associated and to break down or it is break down the RBC is called, it is the very fatal, that is the A antigen or A antibody can be mixed to destroy the RBC. It's called as blood clumping, and that is the agglutination of blood. Okay, agglutination, that is blood clumping. Blood clumping means RBC destroyed, so it is completing. Okay, so if you like my video, please subscribe, share with your friends. Okay.